Hello, everyone. Welcome to today's live broadcast, Deafness in the Genomic Era, Discoveries, Challenges, and Opportunities. I am Kristen Perret-Gentile of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. This event is sponsored by Agilent Technologies. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. The company provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration give people the highest confidence in their solutions. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button, lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credit. I would now like to introduce today's speaker, Dr. Hela Azaiez. Hela is a research scientist at the Molecular Otolaryngology and Renal Research Laboratories, or MORL, at the University of Iowa, USA. She received her Bachelor of Science and her Master of Science degrees in Biological Engineering at the University of Sfax in Tunisia and her PhD in Human Molecular Genetics at the University of Sfax, conjointly with the University of Iowa. After completing a postdoctoral fellowship at the MORL at the University of Iowa, Hela was appointed as Assistant Professor at the Pasteur Institute of Tunis in Tunisia. After working there for 2 years on characterizing the genetic etiology of orphan diseases in the Tunisian population, she returned back to the USA, where she is currently leading the hearing research team at the MORL. Her current research program is focused on the discovery of novel genes, genetic modifiers, and non-coding regulatory elements involved in hereditary deafness, as well as the investigation of genetic contributions to age and noise-induced hearing loss. She is in charge of developing next-generation sequencing platforms and gene panels, along with creating solutions to improve interpretation and clinical correlation of genomic variants. She aims to translate novel knowledge acquired through research into clinical diagnostics. I will now turn it over to Dr. Azaiez for her presentation.
Thank you, Kristen, for this introduction, and thanks everyone for joining us today. I'm very excited to talk to you about some of the projects we are working on at the Molecular Otolaryngology and Renal Research Laboratories at the University of Iowa. Today, I will be presenting our experience with the genetic evaluation of individuals with hearing loss using a targeted genomic panel we have developed in our lab. We will present the challenges we have faced and the solutions we have created to overcome them. We will also show some specific cases to highlight the genetic and phenotypic complexities of hereditary deafness. To start, I would like to give an overview of hearing loss. Hearing loss is the most common sensory defect in humans. It affects 360 million people worldwide, and by 2020, that number is predicted to reach 1 billion. Both environmental and genetic causes have been involved in the etiology of hearing loss. Environmental causes, such as viral infections and ototoxicity, account for around 20% of cases. The remaining 80% are due to genetic causes. This ratio between environment and genetic causes depends on the regional location, where genetic causes are more prevalent in developed countries compared with increased environmental causes in underdeveloped countries. The genetic causes could be actually classified into two categories. Syndromic deafness, which in addition to the hearing loss, there is a defect in another organ or tissue. Usher syndrome, for example, is the association of hearing loss with retinitis pigmentosa. Waardenburg syndrome is a combination of several conditions, including hearing loss and changes in pigmentation of the hair, skin, or eyes. And Pendred syndrome, that is an association between hearing loss and goiter. However, 70% of hearing loss is actually non-syndromic or isolated. And of this, 80% is recessive, whereas 20% is dominant hearing loss. One of the most striking characteristics of hearing loss is that it is phenotypically heterogeneous. It could be sensorineural or conductive, depending on its location in the ear. For example, sensorineural hearing loss is due to defect in the inner ear, whereas conductive hearing loss, the middle ear is affected. Hearing loss is also characterized by the age of onset. It could be prelingual, postlingual, or adult onset. By the severity, which goes from mild to moderate to severe to profound. The frequencies affected could be either low frequency, mid frequencies, or high frequencies. The symmetry of the hearing loss, where both ears could be affected the same or not. And finally, the progression, which is the rate with which hearing loss progress over age. The different combination of all these characteristics gives rise to a wide range of phenotypic outcomes. It's not surprising then that this complex phenotypic landscape is mirrored on the molecular level as well. Over the past 2 decades of genetic research into deafness, a lot of genes have been identified, as you could see here. And the rate of this gene discovery for deafness has tremendously accelerated since the advent of next generation sequencing, with over 150 genes identified to date and over 7,000 reported mutations. An additional challenge in studying deafness is the complex and inaccessible structure where the defect lies, and this is the cochlear. Here is a simplified image of a cross-section of the cochlear surrounded by genes known to cause hearing loss. It's easy to appreciate the diversity of molecules involved, diversity both in function and type, with some involved in ion homeostasis, while others have a more structural role, and others acting as transcription factors. The spatial expression of these proteins is also complex, with some expressed in specific cell types, such as hair cells versus supporting cells, and other genes being expressed as specific compartments within the cell itself, such as stereocilia or mitochondria. This extraordinary phenotypic, genetic, and allelic heterogeneity makes genetic screening of individuals with hearing loss laborious, challenging, and time-consuming. To address this challenge, we capitalized on advances in sequencing technologies to develop a next-generation sequencing platform that we called OtoSCOPE. OtoSCOPE is a targeted genomic enrichment panel that includes 152 genes associated with hearing loss. These genes are linked to non-syndromic hearing loss. All of the non-syndromic hearing loss are included in our panel, but we have also included genes that are involved in some syndromic forms, such as Usher syndrome, Pendred syndrome, and also some common form of syndromic hearing loss like Alstrom, Bohr syndrome, or Wolfram syndromes. OtoSCOPE is regularly updated by adding or removing genes based on new data published. So the next step is actually to perform target sequence capture, and we use the Agilent SureSelect enrichment kit to capture all these exons and make our enriched library. We then use Illumina HiSeq for massively parallel sequencing. As you are all familiar with, next-generation sequencing generates an enormous amount of data that needs to be processed. Over the past 6 years, we have developed and refined a custom pipeline, a custom genetic pipeline, specifically for hereditary hearing loss. This pipeline is comprehensive, and it includes both automated steps coupled with manual curation and expert review steps. So the first step is actually to get the raw sequence data processed, mapped, and aligned to the reference genome. We then do some filtering for quality and for annotation. It's also important to note here that CNV analysis is an integral part of our bioinformatics pipeline. After that, the second step is variant filtering. So we filter variants according to their quality. So we only keep variants with the depth of coverage over 10, present in 30% of the reads, and a QD more than 5. We then look at the functional relevance of the variants. We look at non-synonymous, splice site variants, or indels. We then also, a big part of this pipeline is the minor allele frequency. So we usually end up around 10 variants per sample. These are candidates that we will review in a weekly multidisciplinary meeting attended by clinicians, scientists, geneticists, bioinformaticians, and genetic counselors. And we discuss the phenotypic and genotypic data and see if the genetic data actually is correlated, could be correlated to the phenotype for this specific case. For this, we have actually, in this workflow, we have created and used several key tools which are unique to our pipeline. Two of these tools are the Deafness Variation Database, or DVD, and the second one is AudioGene, use it for audiometric profile. And I will talk a little bit more about these two in the next few slides. So after we prioritize variants, the last part is actually to confirm these variants. So we perform Sanger sequencing, and if all the individuals from the families are available, we could also do segregation analysis to confirm this variant. The last step would be to generate a genetic report that is sent to the clinician. As you could appreciate from here, genetic variant classification is crucial to accurate genetic diagnosis and represents a major challenge in the post-genome era. To address this challenge, we have designed a deafness-specific database called the Deafness Variation Database to provide a continuously updatable, comprehensive resource for variant classification for hearing loss. This database is built on a custom internal bioinformatics pipeline that collects variants and their annotations for all deafness genes. So the gene list is regularly updated by adding or removing genes based on new data. So we review all the genes before including them in our pipeline, in our OtoSCOPE platform. DVD then gathers all information available for all the variants that are present in these genes. And we collect the data from dbSNP, from ClinVar, from AGMD, Exact, EDS, and 1000 genomes. These variants then will go a step of automated annotation and classification. We use dbNFFP to add pathogenicity scores. We use Polyphen, mutation taster, LRT, SIFT, GURP, and PhyloP to annotate for pathogenicity and conservation. An important step here is the minor allele frequency. We use the cutoff of 0.5% for recessive hearing loss and 0.05% for dominant hearing loss. After this step of automated annotation, the variants will undergo actually curated clinical significance. So it's expert panel where we meet and we discuss all these variants in the context of the phenotypic expression in individuals with hearing loss. So all these variants then are stored and they are freely available in the DVD website. To tell you how many variants are present in DVD, that you see it there, it's over 170,000 variants are present in the DVD. Of those, approximately 7,400 variants are annotated and classified as pathogenic, 672 as likely pathogenic. And you could appreciate that the biggest part of all variants in DVD are actually variants of uncertain significance. And this is one of the biggest challenges that not only in the research for hearing loss, but for all other genetic diseases, is those variants of unknown significance. And that's actually what makes the DVD very relevant to what we do. So because it is designed as a deafness specific, and it collates all available data, and it also provides expert manual curation for all these variants involved in disease. So while many laboratories use the publicly available resources such as ClinVar and AGMD to help with variant interpretation, we have noticed that there is some discrepancies between the DVD and both ClinVar and AGMD. In fact, when we compared both the data and classification of variants between ClinVar and DVD, we have noticed that 14.5% have discordant classifications. When we did the same with AGMD, we noticed that almost 5% of variants have discordant classification between AGMD and the DVD. So why is that? One of the reasons and the shortcomings of the ClinVar is that sometimes some variants would have multiple submissions with conflicting classifications. For AGMD, we do know that their data is actually restricted to published papers. So all clinical labs or research labs, you won't have any of their variants in AGMD unless this data was published. And a common remark we have made is that the DVD more commonly downgrades ClinVar and AGMD classification. What that means is for all these discordant calls, actually DVD would say these variants are not pathogenic, they are more likely to be variant of unknown significance, or sometimes they are actually just benign variants. One of also the issues for people annotating and classifying genetic variants is that we try to follow the ACMG guidelines. But sometimes these are hard to follow because they are more generic, so they are not disease specific. And for deafness and for other diseases, there need to be a tailored ACMG guidelines. And I think that now ClinVar is starting expert panels for specific diseases, and which will be really great because we can tailor the guidelines to each specific disease. And most importantly, we will include in those guidelines expert knowledge and curation. So here is just an overview of the variants that are only involved in hearing loss, because some of the genes that are in OtoSCOPE are both involved in hearing loss, but sometimes they are also involved in other disorders. So we only kept the ones that are involved in hearing loss. And we made the comparison between, for example, ClinVar versus DVD. In this table, in this figure, we only put the major categorical changes. What we call major categorical changes is any change into or out of pathogenic and likely pathogenic. So you see the horizontal lines. The red ones are usually getting upgraded from a VUS, benign or likely benign, to likely pathogenic or pathogenic. The blue ones are more downgraded, which are going from pathogenic or likely pathogenic and downgraded to either VUS, variant of uncertain significance, or likely benign and benign. Looking at these two figures, we can appreciate that after expert review and curation, actually more than 300 major categorical changes were made in the DVD when compared to AGND and ClinVar. And this is really, really important to keep in mind. So variant interpretation is actually one component, and we believe that to be able to correlate those variants to the phenotype itself, we need to find a way to check the concordance between, in our case, hearing loss, the concordance between audiograms and variants identified by comprehensive genetic screening in person with hearing loss. So we have developed, to address this challenge, we have developed AudioGene through a long-standing collaboration with the Department of Engineering at the University of Iowa. AudioGene is a supervised support vector machine learning algorithm that uses audiometric data to predict genotypes. For example, the version 4 of AudioGene uses 3,246 audiograms collected from over 1,400 persons with autosomal dominant non-syndromic hearing loss associated with 34 deafness genes. For each of these genes, AudioGene averages all the audiograms in our dataset and group them into 2-decade increments to obtain an audio profile. And we have shown that some genes exhibit distinct audio profiles, making it possible to predict the genetic cause of a person's hearing loss from just entering their audiogram on AudioGene website. AudioGene can also be used to define audio profile surfaces. So audio profile surface, it's a novel method of representing audiometric data that incorporates age as the third dimension. That's the novelty about that. So instead of the usual 2D audiogram, we now have a 3D audiogram, as shown in this figure, representing a hearing loss associated with KCNQ4 gene. In addition to frequency and decibel, age is included to show average hearing thresholds over time. By using audio profile surface, a clinician can provide a person with a hearing loss, specific genetic type of hearing loss, a direct comparison on how that person's hearing compares to expected threshold for that gene at a given age. You see it here with the red line on the audio profile surface. It also helps make an accurate prognosis of the deterioration of their hearing. Using that methodology, we could also make that gene-specific audio profile surfaces. They can also be studied using hierarchical surface clustering to determine whether subclusters provide a better or more appropriate fit for the data. Hierarchical surface clustering was developed as a tool to explore whether novel subclasses for each gene should be considered. For example, we have here two examples. Using KCNQ4, again as an example, hierarchical surface clustering identified two distinct subclusters. One associated with the missense mutation, showing a gently sloping audio profile surface, and the second subcluster associated with truncating mutation, showing a more severe phenotype. Hierarchical surface clustering is also a valuable tool to identify patient subgroups that likely reflect the presence of genetic modifier. In the example shown here, hierarchical surface clustering actually provides two subclusters, 1 and 2, that are not dependent of mutation location or type, or age, or gender. So there is no confounding factors, which actually might hint to the presence of genetic modifiers. Thus far, I have shown you two unique components of our genetic analysis pipeline. However, getting a positive result is more like putting together a puzzle. We have to take into account different pieces in order to establish a holistic and comprehensive view of the genetic results. We have to include the variant frequency, variant type, that has to segregate in the family if there are more individuals available. The phenotype making a phenotypic correlation, pathogenicity predictions, and finally, if there is any functional data available for this variant. So using the method I have described, we have analyzed over 5,000 samples to date. We use the combined knowledge provided by both the positive and the negative cases. So positive cases are the cases where we were able to identify the genetic mutations in known deafness genes. Negative is no diagnosis could be made. One important thing is here I'm going to share with you the data we got for screening all these cases. The overall solve rate is around 50%, but it is hugely influenced by several factors. One of them is the inheritance mode, with autosomal recessive cases showing a higher overall solve rate than autosomal dominant. The other thing is ethnicity. And you could appreciate that cases with African descent have the lowest solve rate amongst all populations studied. And that is still true, and it's actually a good motivation to study this population more in order to identify novel genes. At the other spectrum, we could see that populations where consanguinity is culturally accepted, the solve rate is the highest. And that is due mostly to homozygosity, because these consanguineous marriages usually produce homozygosity, and the solve rate is really higher, and sometimes it's around even 100%. The genetic also we have shown that some genes are more prevalent than others. Here, this is the average general gene prevalence. And I would like to emphasize here that for all the patients I am talking about, we have excluded the ones that were GJB2 positive, which meaning that the ones we have found a mutation in GJB2. We did that because most of the samples we get have already undergone GJB2 testing, and they were negative. And we thought that including them, including the patients with GJB2 mutations would skew kind of the results and the statistical analysis. So we just excluded them from this, from the rest of our analysis. So we could see that SLC26A4, MYO15A, cadherin23 are the most prevalent genes, and that have already been shown before. The interesting part here is the spectrum and prevalence is actually also ethnic dependent. And you could appreciate here how each population has a different gene prevalence. One also that we were interested in is to see the variant type. And we saw that for both recessive and dominant, most of the variants identified are novel. And for example, for dominant, only 20% of the variants that are identified in patients with hearing loss, with autosomal dominant hearing loss, are only 20%. Only 20% of them are already known pathogenic. And to add another layer of complexity, of all the variants that are identified, almost half of them are variants of unknown significance. And that's actually what makes things, as we talked about it earlier, really complicated. We have also seen that missense mutations are more prevalent in dominant, and that nonsense and truncated mutations are more prevalent in recessive hearing loss, which we have expected before. What I would like to get your attention to is the CNV analysis here, and which until now is like till the advent and implementation of next generation sequencing CNV analysis was very costly and time-consuming to screen for. And I am going to show you a few more examples of the importance of CNVs. For example, we have shown that CNVs can be found in about 20% of deafness-causing genes. And most of the CNVs are actually whole gene deletions, so the whole gene is deleted. We have also seen duplications and conversions. Most of the time, those CNVs are homozygous. They could also be heterozygous with another single nucleotide variant on the other allele. So with all these positive cases, there are a few things that we can learn from. And one of the most important things that is relevant not only for research but also for diagnostics and for genetic counseling is genotype-phenotype correlations. And depending on those genes and depending on those variants and their phenotype, we could actually show some genotypic complexity. We could expand the phenotype of some of the genes. For example, they were shown before to only be associated with dominant versus recessive or syndromic versus non-syndromic. One of the important things we can actually also study is genetic modifiers. And in the next of my slides, I am going to go through each of these three categories. So we will start with the genotypic complexity. And our first case is a 5-year-old Irish female with congenital, severe to profound sensory neural hearing loss. The family history showed an extended family with hearing loss and a mother with white forelock. For those of you who are not familiar with some of the genetic of hereditary hearing loss, a white forelock or a problem of pigmentation in association with hearing loss is a strong indicator of Waardenburg syndrome. So this sample, this case was referred to us after a negative result from another institution. So they ran a Waardenburg panel test and it came out negative. When we ran this sample in our pipeline, we couldn't find any single nucleotide variants in any of the genes associated with Waardenburg syndrome. But what we found is actually a heterozygous 2-exon deletion in SOX10. So we actually solved this case because of the presence of the CNV. And that actually really highlights the importance of integrating CNV detection in any next generation sequencing panel for hearing loss. Our second case is also to highlight the importance of CNV. So this case is a 6-month-old with mild to moderate bilateral non-syndromic hearing loss and negative family history. So our CNV analysis indicated a homozygous deletion of the stereosilin gene. We could have stopped it there because that would explain their hearing loss. However, one of the interesting characteristics of this genomic location of stereosilin gene is that it's a duplicated region. And you could see here that stereosilin lies next to Caspr2 and that several 100 kbs later, there is a pseudogene STRC-P1. And when our analysis pipeline actually also screens the region, this whole region, including stereosilin, Caspr2, the pseudogene. And what we have seen there is that it's actually not a deletion of stereosilin, but rather a gene to pseudogene conversion through a non-allelic homologous recombination. That was really amazing that because before we just assume it is just a deletion of stereosilin, where here we are shown actually this whole region is prone to genomic rearrangements, and in this case there is a conversion from the gene to the pseudogene. So here is another case. Again, we see a homozygous deletion of a stereosilin in a 1-year-old male with mild to moderate hearing loss and a family history that is negative. So here, again, it's the homozygous deletion in stereotypin. The pseudogene was normal when we screened it. So that could have been DFNB16, which means it's non-syndromic hearing loss. However, our pipeline also screens for CASPR2. And what you see here is actually also a homozygous deletion of CASPR2. Why this is important? That is actually very important because truncating mutations in CASPR2 have been associated with male infertility. So homozygous deletion of stereotypin and CASPR2 actually would give rise to deafness and fertility syndrome. So this boy is going to be infertile, which is really very important for the family to know and for genetic counseling later. We have shown till now a whole gene deletions or a partial gene deletions, but we need also to emphasize the importance of single exon deletion. That is the need for a high resolution CNV detection. In case 4, this is an 11-year-old female with congenital profound sensory neural hearing loss. She was adopted, so we don't have a family history. But she has heterochromia, iridium, white forelock, and slight brachydactyly. We did OtoSCOPE, and we found that there is one single exon deletion. This would have been completely missed if it was done by Sanger sequencing or if we just screened next generation sequencing but without CNV analysis. The next case is actually also really interesting. This is an 8-year-old male with congenital moderate sensory neural hearing loss, negative family history, and the physical exam only showed myopia. And this is their audiogram. So when we performed OtoSCOPE, we found actually a homozygous stop mutation on AudioGene. This is already a known pathogenic mutation. So this is great. So this person is actually carrying a homozygous stop mutation. The story could have ended here. However, when we did CNVs, we actually detected a heterozygous deletion in AudioGene. So that variant we saw and we thought is homozygous is actually a hemizygous. And the etiology and the genetic cause for the phenotype in this case is actually compound heterozygosity for one stop mutation in one allele and a deletion in the other. This is completely would change genetic counseling and carrier screening, and it's really important. One of the other also challenges and we have encountered, and some actually also of the cool cases, is genetic heterogeneity in one family. For example, the families of Iranian descent, segregating severe to profound non-syndromic hearing loss, and exhibiting several loops of consanguinity. So we sent one individual for OtoSCOPE. The results came out with a homozygous mutation in cadherin 23. So great, we did segregation analysis. Turns out, to our surprise, this variant only segregated in one branch. The other branch, none of them even carry this variant. So we sent another patient from the other branch to screen by OtoSCOPE. We found actually homozygous myo7A mutation. And then when we did the segregation analysis, it turned out that this mutation only segregated in the other branch of the family. There is only one individual who both was homozygous for both mutations. And that is actually also really important to screen when it is a big family. It is better to screen a couple of individuals, just especially from different branches, just to make sure we are not missing anything. With this case, we are going to add another layer of complexity. So this is an Iranian family with Usher syndrome type 1. That's the most severe kind of Usher syndrome. We sent this person for OtoSCOPE, and it was negative. So we said, oh, this is a great family then for whole exome sequencing to identify a novel gene, because we thought none of the known genes explained the phenotype in this family. So we sent this patient and actually 2 others for whole exome sequencing. To our surprise, we found out that actually the 2 other patients actually carried 2 known pathogenic mutations in H1C. This H1C has been already linked to Usher syndrome type 1. This individual actually did not have none of the variants. We contacted the clinicians, and then they went back and checked, and then it turned out that this patient had only some kind of eye disorder that they called retinitis pigmentosa. So completely changing the phenotype for this patient. When we analyzed this patient's whole exome sequencing by himself, we actually discovered a mutation in CACNA1F, and that is X-linked, and mutation in CACNA1F have already been involved in congenital stationary night blindness, that is kind of RP-like, or chondrodystrophy. So for this actually family, there are 2 distinct phenotypes and 3 distinct genes in one family, because the 3rd generation, the affected girl in the 3rd generation, we still could not explain her hearing loss. It might be coming from her father because he's a married in. But that is just to highlight some of those complexities we are talking about. Another interesting case is we got this 43-year-old male with bilateral profound hearing loss and known family history, and the clinical diagnosis was actually Usher syndrome. But when we performed genetic testing, we found that he is actually carrier for 2 pathogenic mutations SLC26A4. That explains his hearing loss, and another mutation in OPA1, which explains the optic atrophy. So actually, we changed the diagnosis completely from Usher syndrome to just non-syndromic hearing loss due to SLC26A4 and optic atrophy. The second class of things we learned from genotype phenotype correlation is phenotypic expansion. And I'm going to talk about this family where we have identified a mutation, canonical splice site mutation in COL11A1, segregating with the disease in this autosomal dominant non-syndromic hearing loss. So what we knew before is that mutation COL11A1 have been only linked to Stickler and Marshall syndrome. This is the first time that mutation in COL11A1 have been associated to autosomal dominant non-syndromic hearing loss. The second case is with PTPRQ. PTPRQ has, to date, has only been linked to autosomal recessive non-syndromic hearing loss that is severe to profound. In the family, we found it associated with autosomal dominant non-syndromic hearing loss, and since recessive mutations are null alleles or lack of function, so we predict that the mode of mechanism of PTPR dominant mutations in PTPRQ are gain of function or dominant negative. We have also shown that PDZD7 is actually causative for non-syndromic hearing loss, because till now PDZD7 has only been shown to be just a modifier for the retinal phenotype in ASH2. So all these are really important for, again, for diagnosis, for genetic counseling, for carrier screening, and just even to shed light in some of the complexities and of hearing loss. So the next part, I'm going to talk about genetic modifiers. And here is a family that segregates autosomal recessive non-syndromic hearing loss, and we found 2 mutations in TMC1 gene. It segregated in the family. The only interesting thing is 2 patients exhibit a more severe phenotype than the 2 others. So 2 exhibit only a high frequency hearing loss, whereas the 2 others exhibit a profound hearing loss. So here we suspected there is the intervention of a genetic modifier. But could this genetic modifier, is it making the the phenotype worse? Is it actually protecting? We went back to the literature to look through literature, but also through some of the cases we had before. And we noticed that those mutations have been linked to severe to profound hearing loss in other patients, which actually suggesting that what we are looking for here is a genetic modifier that would protect from the full effect of TMC1 mutations. So hypothesis for this outlier phenotype is it could have been also due to fortuitous mutation in a second gene, but we have already checked for that by performing OtoSCOPE and genetic screening in all these patients, but we couldn't find any variant in known deafness causing a gene that would segregate with the outlier phenotype. The other hypothesis, as I said earlier, is the genetic inheritance or a genetic modifier, and for this we plan on performing whole exome sequencing and whole genome sequencing and study the pathway and interactomes of protein that would interact with TMC1 or modulate its effect. So what is next? So I have talked about the positive cases and how they actually provide a better understanding of hearing mechanism and how they open many doors. But what about the negative cases? The negative cases are actually really interesting because these patients and cases are great candidates for novel gene discovery. There are several ways and several labs use different approaches. We do use a tiered approach. For those negative cases, we would screen them with another targeted genomic panel for candidated genes that we have developed in the lab. These genes are these genes are hair cells or enriched, and the mouse models for some of them are deaf. So these are great candidates. Patients that are actually also negative for hearing panel, and we will go to perform whole exome sequencing. And using that strategy, we've actually identified several genes using this approach. But as you may expect, and as you may know, a lot of these cases, a lot of the cases still remain unknown and unidentified. And for those, we could again use targeted panels if we would like to look for regulatory elements. We could also use whole genome sequencing. So there are different approaches and different ways, but they all actually open doors for research, identifying novel genes, identifying regulatory elements, identifying genetic modifiers. Our pipeline actually allows us to collect such really interesting cohorts for further studies and research. So as a conclusion, I would like just to emphasize a few points here. That targeted gene panels, rather than exomes or genomes, for deafness offer an efficient, cost-effective, customizable, high throughput, and high depth sequencing approach to detect rare variants and CNVs. Speaking of CNVs, their detection and high resolution detection must be integrated in all next generation pipelines for deafness. It is also really important to integrate the audiometric and the genetic data to facilitate interpretation of genetic variants. Targeted gene panels could be used as front-end filter to generate a carefully selected study population enriched for hearing impaired individuals likely to carry mutations in novel genes or non-coding variants in regulatory regions. I would like to thank all our collaborators all over the world for their help and great input. I would also like to thank all individuals and families who participated in this research. Would like to thank Richard Smith, the director of the MORL, Kevin Booth, graduate students in the lab, and also all people and team members at the MORL for their magnificent work and tireless, really unbelievable work we are doing here. I would like also to thank Thomas Gazavant, Terry Brown, and Michael Schneider from the College of Engineering at the University of Iowa, and Hussein Najmabadi and Kimia Kahrizi from the University of Social Welfare and Rehabilitation Sciences in Iran. All these people's help has been invaluable and has advanced our knowledge of the genetics of hearing loss. I would like to thank you for your attention, and I would be happy to answer your questions. And thank you again for attending today's webinar.
Thank you for that informative presentation, Dr. Azaiez. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, looking at OtoSCOPE versus whole exome sequence, which one is a better option and more reliable?
This is a great question. Actually, we receive this question a lot. And in our experience, we think that OtoSCOPE or targeted, custom targeted panels outperform whole exome sequencing because first, the OtoSCOPE targeted panel is actually deafness specific. So included in there is all, besides all the coding exons, there is also variants that are in non-coding regions, either antronic or intergenic. We could also tile for genes for CNV detection, and this is not possible with whole exome sequencing. The other thing is OtoSCOPE, because of the great depth of coverage, we could actually accurately detect rare variants. This is, the accuracy is not the same with whole exome sequencing, and we have done several studies, and we have shown that the detection rate with OtoSCOPE is way greater than whole exome sequencing. Some other benefits of using targeted panels are also the high throughput and the decreased cost. Because, as you know, whole exome sequencing is more expensive, and it's not as high throughput as targeted panels. Also, one other level of argument for the use of targeted panels is the ease of analysis, because here we are only looking at deafness-specific genes. So that makes the analysis easier, more relevant, and it also kind of resolves the issue and the challenge and the burden of incidental findings and what to do with them. Any other question?
Okay, great. Yes, we have another question. How do you confirm the CNV?
This is another actually great question. And we have done several studies before, and we confirmed our CNVs by MLPA. We have also done an independent study to confirm our analysis detecting CNVs by whole genome sequencing using array comparative genomic hybridization. We also, when it is possible, we Sanger sequence. We do segregation analysis, or we Sanger sequence the breakpoints. Thank you.
Okay. Let's see. For the next one, in your opinion, what explains the significant number of unsolved cases?
So the number of unsolved cases, it's around 50%, and again, depending on the population and ethnicity and studies. But there are several main reasons for this high rate of unsolved cases. First amongst them is we can only screen for genes for known genes. So the unsolved cases, actually, a lot of them would be accounted for by novel genes yet to be identified. Another reason is we are not actually screening for non-coding variants in regulatory elements or even, that could be entronic or heterogenic. So we are missing another whole class of variants, which are the non-coding ones. Another reason why is also the unrecognized genotype-phenotype correlations. I have shown here a couple of examples with PTPRQ and COL11A1. And for example, if it was a diagnostic lab and they have autosomal dominant family, and then they found a gene, in our case, a nonsense mutation in PTPRQ, they would not suspect that this is the cause of this family hearing loss. Because first, it is a recessive gene, and second, it was a nonsense mutation. So this unrecognized genotype-phenotype correlation actually accounts for, we believe, an important, significant proportion of unsolved cases. Another level is also the synonymous variants, because no one reports synonymous variants because we do not believe they are disease-causing, but these variants, actually, some of them could be or could have an influence and it could impact splicing, and that they could be also, they could account actually for some of the unresolved cases. Thank you.
Okay. Our next question we have, what targeted capture is the OtoSCOPE based off of, and how did you choose that technology?
So we designed OtoSCOPE using Agilent, SureSelect, custom targeted capture. So why we used Agilent? Because I think one of the main reasons is first the design flexibility. We could actually put anything we would like in our platform. It's custom, it is personalized, so it could be coding exons. We put in there promoter regions. We put sometimes up the single nucleotide variant, that is intergenic, because we were interested in those variants. So the flexibility in the design is a really good, is a really important factor. Another important factor is the capture efficiency. We have had great success rate using the SureSelect custom capture because of its efficiency. One other reason is also the ease of the experimental design, because it is fast, but it also could be automated. And as you saw, we have already sequenced over 5,000 samples. So automation and the ability to automate sequencing is a big factor in our decision. Thank you.
Okay. Let's see the next question we have. There are a large number of discrepancies between DVD and HGMD and ClinVar. What do you attribute this to?
So ClinVar, ClinVar and HGMD are both great tools and great databases, and they give a phenomenal help. But they do also have a couple of shortcomings. With ClinVar, the fact that one variant could have multiple submissions because several labs put in their own interpretation and classifications, and some of the time they are conflicting. So it is really hard to know which one is correct. Sometimes for the same variant, it could be benign or pathogenic, and then it's up to the researcher or the clinician to figure out which one is correct. Also, anyone can actually deposit in ClinVar. So that also adds another level of kind of challenging. For HGMD, their variants are only based on published literature. So if a variant has not been published before, you will not find it in HGMD. Also, sometimes the papers referenced or that HGMD is based on, sometimes those papers are a little confusing and information is missing. regarding if it actually is disease-causing or not. So they just report variants, and sometimes they are not really clear if this one is, for example, was found in trans with another mutation or it's just by itself. So reading those papers might be misleading, and that's actually what accounts for those discrepancies. For the DVD, since we do beyond the automation step that classifies all variants, we actually also expert curate all of those variants. So we correlate them to specific phenotypes because we have seen them in one of our patients. So it's like this is over 5,000 patients, so we have seen a lot of variants. And it was possible for us actually to make a correct call regarding those variants. Thank you.
Thank you. All right, the next one we have. What if the whole exome comes back with rare mutation of USH gene? What is the accuracy rate when the OtoSCOPE is negative?
So as we saw here, if this variant is in a known deafness gene, all bets are we are going to detect it using OtoSCOPE. The only challenge here, and I presented one of the cases here with the ASH1C, and you saw that because of the lack of phenotypic characterization, so the clinical information we were getting was not accurate, and that led us to actually not being able to find the mutation. But when we did whole exome sequencing, we found a mutation, but we found it in another gene that has never been linked to just deafness. So a short answer would be using OtoSCOPE, we will find any mutation if it is in a known deafness gene. If the clinical was not accurate to begin with, it is hard. Even if you use exomes or any other kind of screening, you won't be able to find the correct genetic code. Thank you.
Okay. What targeted capture is OtoSCOPE based off of? How did you choose the technology?
Again, it is based on SureSelect, custom targeted capture. We use it because it was one of the best and leading companies in this domain. We also use, actually, by the way, we also use SureSelect whole exome sequencing. So we use both the custom target capture and the whole exome sequencing target capture, but for two different research purposes. We always follow a tiered approach in our analysis. So we always screen all patients and all cases that we receive using OtoSCOPE, the custom targeted panel. And then for everything that is negative, we couldn't find and identify any genetic code for their hearing loss using OtoSCOPE, we will send them, we will perform whole exome sequencing using the Agilent SureSelect kit too. Thank you.
Okay, it looks like we have time for one more question. Now that we know the genetic makeup, can we do anything nutritionally or medically to change the hearing loss?
Yeah, this is one of the, that's what we all aim to actually, all the basic research and genetics. Our final goal is actually to develop therapies. And there are several studies that have been made, and a lot of them are based in gene therapy. Some people are using the siRNAs, and some people have done it also for Usher syndrome, and I think it is promising. And again, before any therapy could be developed, we really need to characterize the genetic makeup, but also for both recessive and dominant, because the therapies would be different, if it was dominant or recessive. And so that is nutritionally, there are few studies. I'm not going to go there, but medically and in the gene therapy, there are a lot of things going on. Thank you.
Okay, wonderful. I would like to once again thank Dr. Azaiez for her presentation. Do you have any final comments?
No, thank you. I would like to thank everyone for joining us. It has been a real pleasure, and please feel free to send me any questions because of the time here, the time limit, but please feel free to send me any question. We will be really happy to help answer any of your questions and provide more information. Thank you very much.
Great, thank you so much. Today's webcast will be available for on-demand viewing through November of 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. And we'll see you next time. Goodbye.